Alright, so let's get started with chapter 13. So the first section is about hormones, which transmit signals among different cells from different organs to each other. So first, let's just start off with what is a hormone? Yeah. Yes, it is a signaling molecule. And are the types of signals transmitted by a hormone short distance or long distance? Yeah, long distance. And so this tends to mean between different types of organs or tissues. So there are two sort of classes of hormones that are defined by how they travel through your body. So one type of hormone, or can somebody tell me what is one mode of travel for hormones and what that's called? Yeah. Yeah, through the blood is one. And what do we call that? That's endocrine, yeah. Oops. And what is the other way of travel? Yeah, through ducts. And what is that called? Yeah, exocrine. Great. So we all know what hormones are. It's a good start. So so let's start talking about different categories of hormones and their functions. So everybody take a couple of minutes and start matching up the functions of the hormones and the category or specific hormone on the right. So let's get started with number 1. Which hormone is involved in fight or flight, blood vessel contraction, heart rate, fuel metabolism, all that good stuff? Yeah, it's C. Epinephrine and norepinephrine. What about regulation of salt and water by the kidneys? Yeah. B. Yeah. The mineralocorticoids. And this is kind of in the name, right? Minerals, salt. Maybe that'll help you remember. All right. Number 3, released when levels of blood glucose are low, promotes breakdown of glycogen and synthesis of glucose in the liver, promotes breakdown of fats in adipose cells. Yeah, it's glucagon. One way, it's kind of silly, but I like to remember glucagon because it kind of just sounds like all the glucose is gone, which is when it gets released. So yeah, now you'll never forget what glucagon does. All right, so number 4, released when levels of blood glucose are high, promotes synthesis of glycogen, protein, and fat by muscle, liver, and adipose cells. A, yeah, that's insulin. And insulin and glucagon, as you can kind of see, have opposing functions. When glucagon is signaling, it's starting one set of pathways. When insulin is being released, it's sort of doing the opposite of what glucagon is trying to promote. And we will talk a lot more about insulin and glucagon signaling as we go through the next set of chapters, because they are very involved in glucose metabolism, which is what we'll be discussing in chapters 15 through 18. Or mostly in 15, 16, and 17. Anyway, doesn't matter. So let's look at number 5. So which one of them influences carbohydrate, protein, and lipid metabolism, inflammation, and the stress response? Yeah, glucocorticoids. Great. Any questions so far about the general hormone functions? Great. So let's talk about the pancreas. So what type of gland? So yeah, pancreas has 2 types of glands. Which one is the larger part of the pancreas? Exocrine, yeah. It's a really tiny space, my bad. Exocrine. And what does the exocrine gland produce? Yeah, digestive enzymes. And what kind of gland is the other small portion of the pancreas? Endocrine, yeah. And what does the endocrine gland produce? Sorry? Yeah, polypeptide hormones. And a lot of those polypeptide hormones are used for controlling blood sugar levels. So which of these hormones are produced in the pancreas? Yeah, insulin's one. Yeah, and glucagon. Great. And are those from the endocrine or the exocrine portion? No, it's not. Yes, it is endocrine. Yeah, so these are polypeptide hormones that are used to control blood sugar levels. And does anybody remember what type of cells make insulin? So, yeah, it's beta cells. And what about glucagon? It is alpha cells. Yeah, you got, just pick a Greek letter, you got it, yeah. Awesome. Any questions about pancreas? All right, so let's move on to the adrenal gland. What are the two main parts of the adrenal gland? You can just name one. You don't have to name both if you're shy. Yeah, the medulla is one. And what about the other one? Yeah, the cortex. All right, so now which of these hormones are produced in the adrenal glands? Yeah. B and E, you said? Yep. And any other ones? And D. Yeah, so the ones that are not produced by the pancreas in this list are produced by the adrenal gland. So maybe a harder question is, which ones are produced by the cortex? Yeah, B and E. And that's kind of also right in the name for you. So it's a corticoid, so corda, son of cortex. They are made in the cortex. And they're also, these steroids in general are called adrenocorticosteroids, right? So they're made in the adrenal gland, in the cortex, and they are steroid hormones. So it's helpful to break apart those names. So that leaves catecholamines. Where are those made? The medulla, yeah. And what type of hormone that we have talked about are catecholamines? We mentioned them earlier in these slides. Yeah, epinephrine and norepinephrine. Great. Sorry, that's a really messy way of, I wrote nor in parentheses and then epinephrine because I didn't want to write it twice. Any questions about the adrenal gland? Cool. Okay. So that brings us to the next part of this chapter, which was about signal transduction, which is, so hormones were taking signals between cells through your body. Signal transduction is all about getting those signals inside your cells and telling your cells what to do. So click on there. All right, so let's start thinking about signal transduction pathways. So what are the two major categories of transmembrane receptors that we see in signal transduction pathways? Yeah. Yeah, so we have RTKs and GPCRs. So does anybody know what RTK stands for? Yeah. Yep. So we have receptor tyrosine kinases. And what does GPCR stand for? Yeah, G protein coupled receptors. So their names already tell you a lot about what they do. So receptor tyrosine kinases, they are transmembrane receptors. They have a tyrosine kinase domain that happens to be on the intracellular portion, meaning that they add a phosphate group to a tyrosine amino acid. So those three pieces of the name will tell you important things about what it does. Same idea for GPCRs. So GPCRs are receptors that are transmembrane receptors, and they are coupled with a G protein that is on the intracellular portion of the receptor interacting with it. And they are coupled, meaning that when the receptor is activated, that gives a signal to the G protein. Okay, so let's fill in the next blank. So signaling molecules or effectors bind to the Witch domain of the receptors, which will undergo a conformational change to initiate a signal transduction pathway. Yeah. Yes, extracellular. So this is where things like hormones come in. Hormones are traveling through your bloodstream. They're outside of cells, but they can also bind to receptors on the extracellular portions of cells to give information to that cell that can eventually get sent inside the cell. So that brings us to the last part. So many transduction pathways involve the addition of phosphate to enzymes by which type of enzyme? Yeah, kinases. So a lot of signal transduction pathways are just long chains of enzymes phosphorylating their substrate, which is another enzyme that will phosphorylate its next substrate, and so on and so forth for many steps. Any questions about signal transduction pathways in general? All right, so let's talk more specifically about receptor tyrosine kinases. So what conformational change do we see that take place in RTKs when the effector binds? Yeah, dimerization. So when there's no ligand or effector bound, they are just monomers hanging out in the membrane. When the ligand binds, two RTKs will come together to form a dimer, which is the answer to this question. And so how does dimerization help to actually activate a signaling pathway? Go for it. Yeah, autophosphorylation. So this is where the tyrosine kinase part of the RTK comes in. So each one of these RTKs has a protein tyrosine kinase domain, and they will phosphorylate each other, which is often referred to autophosphorylation or transphosphorylation. And then once they are phosphorylated, that is basically a binding site for other molecules to float in and start activating their own particular signaling cascade that, like I mentioned, usually involves more phosphorylation. And if anyone wants to look at a picture of that, that is somewhere in these slides. So receptor tyrosine kinases, they start out as monomers before the ligand binds. Once the ligand binds, they come together as a dimer, and that helps to activate the protein tyrosine kinase domains, which are shown as these little semicircles here with the little radiation coming out. That means they're activated. And then once they're activated, they phosphorylate each other. So phosphate groups get added on to each other by themselves. And then once those tyrosines that are now have phosphate groups attached to them, other signaling adapters and kinases can come in and bind to those phosphorylated tyrosine residues. Any questions on how that works? All right. Where are we? Yeah. So now we're up to G protein coupled receptors. So what conformational change takes place in G protein coupled receptors and G proteins once an effector binds? Do you want to guess or say something? You look uncertain. It's okay, you can guess. So, yeah, so you're right that there is movement happening, but that's more about kind of the next part, like how it activates the signaling pathway. So in terms of conformational changes, so is the change happening in the GPCR itself or with the G protein? The G protein, yeah. And so we know, like you mentioned, G protein is heterotrimeric. It has an alpha, a beta, and a gamma subunit. So something is going to happen to one of these subunits. Anybody want to guess which subunit? Yeah. Yeah. Yeah, exactly. So in the G alpha, there's an exchange GDP for GTP. And this essentially sort of activates the G alpha portion of the G protein. And what happens as a result of the GTP bound G alpha, or what happens to that G alpha? Yeah. I mean, kind of. It's less so within the protein. It more is about how it's interacting with some of the other things we're talking about. So, yeah, exactly. So at first, the G, alpha, beta, and gamma are all mushed together. They're, besties. But once the G alpha is in the GTP form, it starts to dissociate from beta and gamma. So how does this help to activate a signaling pathway? Where is the G alpha going? What's it doing? Yeah. So that's true for the AC pathway, but for GPCRs in general, the AC enzyme is not always the next step. But yeah, you're sort of thinking in the right direction. The G alpha is sort of before it even gets to the AC. How is it getting there? What's it doing? Where is it going? Is it like detaching from the membrane and like floating around the cell? What's it doing? So it attached. So let's take a picture. Let's take a look at a picture of it. So we have this is sort of before everything starts, we have our inactive G protein. We have our G alpha subunit with GDP bound, and it's hanging out with beta and gamma. Once the signaling molecule starts to interact with the receptor, then we have this activation shown with those little radiant things. The GDP leaves, the GTP comes in. G alpha has GTP form, is in the GTP form, and beta and gamma are over here. So the whole time, right there, already, they're in the membrane. So these are lipid-linked proteins. They have little lipid tethers that help keep them attached to the cell membrane. So what type of movement are they capable of? Are they stuck in the cell membrane? Can they leave and come back? Can they move in any other direction? Yeah. Go for it. Exactly, yeah. So if they're tethered to the membrane, but they're very capable of lateral diffusion. So that's what happens. So once the G alpha dissociates from beta and gamma, it starts to laterally diffuse around the membrane. And that will eventually help it start a signaling cascade once it finds its target, which is somewhere else in the cell membrane. So the target is always an integral membrane protein. So any questions about GPCRs in general? Some faces being made. I apologize. All right, so let's start talking more specifically now about the AC pathway. So we just talked about GPCR pathway getting activated in general. Ligand binds GPCR, G alpha loses GDP, gains GTP, starts moving around in the membrane. So in the AC pathway, what enzyme is the target of that G protein that's going to get activated? I'm sorry? Oh, interesting. Okay. So, what enzyme is being activated by our G protein? Yeah, adenylyl cyclase. And sometimes you'll also see it written as adenylate cyclase. So don't get confused if you see adenylate or adenylyl, it's the same thing. And that's also what AC is short for. So you can also call it AC. So this enzyme, the AC enzyme, converts what into the secondary messenger? Yeah. Yeah, exactly. ATP is getting converted into cyclic AMP. So what is a secondary messenger? Yeah, exactly. So a secondary messenger is a small molecule that's made inside the cell as an intermediate in the pathway. So it's different from the primary messenger. The primary messenger is the first messenger, like the ligand that activates the pathway by binding to the extracellular region of the receptor. The secondary messenger is made inside the cell as a response to the cascade that's started by the primary messenger. And so the secondary messenger can help to further activate later things in the pathway, and it also can help to amplify the original signal, because if you make a whole lot of secondary messenger as a response to just a few primary messengers, suddenly you have a lot more signal going on saying to activate your pathway. So this product, cyclic AMP, activates, yeah. I believe so. I guess for these types of pathways that begin outside the cell, I guess for like steroid hormones, they just float through the cell themselves, and I would also consider those a primary messenger. So not always, but they originate from the outside. Yeah. So cyclic AMP, what enzyme is it activating? Yeah, PKA. And what does PKA stand for? Yeah. Yeah, it's protein kinase A. So it has kind of a very generic name, just protein kinase A. But, it tells you some things about it. It's a kinase, so what is it doing? Yeah, it's adding phosphate groups to its substrates, which are downstream in the signaling pathway. And we're going to see PKA again, or we've kind of already started to see PKA in a way, because when we were talking about glycogen phosphorylase, remember, is activated by the enzyme phosphorylase kinase. Phosphorylase kinase is activated by PKA. So if you go one step further back, PKA is what's going to activate that. And ultimately, what is controlling it, the primary messenger here, is the hormone glucagon. Glucagon is going to activate the AC pathway, which is ultimately going to activate PKA, which is going to activate phosphorylase kinase, which is going to activate glycogen phosphorylase and help release glucose, because glucose levels are low. Yeah, so let's start talking a little bit more about how that works. Sure. Yes, it is. Yeah, sorry. Sure. So let's talk a little bit more about glucagon signaling. So what is glucagon binding to initiate the signaling pathway? Yeah, a GPCR. And so what are some of the outcomes in the liver in response to glucagon signaling? So remember, glucagon is an indication that energy levels are low. There is a lack of glucose in the bloodstream, in the environment. Yeah, so we want glycogen breakdown to free up some glucose because we need more. And what do we do in terms of glucose? Synthesize or break it down? Yeah, we're going to synthesize it. So once again, there's not enough glucose. You can't break it down because there isn't any. You want to make some, or you need some, you got to make some. So glucose synthesis. So then once glycogen is broken down and glucose is made, is glucose going to be imported or exported from those cells? It's exported. Yeah, once again, there is no glucose there to import. You're making it in the liver so you can send it out to other organs where it's needed. So that's the liver. So the next question is about muscles. So fun fact, there are no glucagon receptors in muscles. So even though in general a hormone would act the same at liver and muscle, in this case, muscles don't have a way to listen to what glucagon is telling them. So we wouldn't see a response to glucagon in the muscles. But if we could, it would be the same as in the liver. questions about glucagon signaling? All right, so let's go on to insulin. So what kind of receptor does insulin bind to? What was that? RTK, yeah, to initiate a signaling pathway. So we talked a little bit about for glucagon how it activates the AC pathway and PKA, and it's sort of relatively straightforward in terms of getting to glucose. For insulin, it's much more complex. Insulin can activate several different pathways that have a sort of variety of effects that is more complicated than we're going to get into for this class. For the purpose of class, it's enough to know that insulin binds to an RTK. Yeah, so what does it mean, right? When there's insulin signaling, is there a lot of glucose or a little bit or not enough glucose available in the bloodstream? Yeah. There's a lot of glucose. Yeah. So the outcomes in the liver due to insulin signaling would be, what would we do for glycogen? Synthesize or break it down? Yeah. Exactly. Yeah. So if there's a lot of glucose available, there's no need to break down glycogen because there's already plenty of glucose. It's a good time to start building up glycogen to save it for later when there's not enough glucose around. And I think somebody already said it, glucose is going to be imported. There's a lot of glucose in your bloodstream. You had to bring it into the cells so it can be stored for later. Or used, I guess, if for other cells. For the liver, it's for storage. For other cells, it could be for usage. So what does that mean for muscles? I mean, liver is going to use it too. I mean, it's not like there's not glycolysis in the liver, but the liver is like one of the main places where glycogen is stored in your body. So I tend to think of it more as a storage, but it's not exclusively for storage. So what about in the muscles? Glycogen is going to get synthesized or broken down in response to insulin. Yeah, it's not a trick question. Insulin does the same thing. Yeah. So insulin is a sign that there's a lot of free glucose around. So your muscles are also going to start building up glycogen so that when they need glucose quickly, when you're contracting them, it's there for later. And so same idea for glucose itself. You're going to import it or export it from your muscle cells. You're going to import it, yeah, because you want to bring in that glucose so that you can either turn it into glycogen or use it. So let's start thinking about a little bit one of our other hormones that we didn't discuss specifically. So we talked a little bit about glucagon and insulin and how they have sort of opposing functions. So where would epinephrine and norepinephrine fit in? Would they be more like insulin or more like glucagon? Yeah, so we talked about how glucagon signaling is a sign of low blood sugar levels. It's a time when you're going to be breaking down any stored glucose or making more glucose to use, versus insulin signaling, which is a sign you have high energy levels and you're going to mostly start bringing in insulin or in glucose for storage. So when you think about epinephrine and norepinephrine, which one would that resemble more? Would they tell your liver and muscle to store energy for later or to start breaking it down and using it? Yeah, exactly. So norepinephrine and epinephrine are there to help mobilize energy resources for your fight or flight response. So it's there you're gonna, start breaking down glucagon, freeing up glucose, doing glycolysis, because you need to run fast and contract muscles and all that stuff, and dilate blood vessels and everything else too. yeah, so like I sort of said earlier, there's a lot here, but a lot of it is looking at the same types of factors. We're going to be looking at glucose and insulin a lot. We're going to be thinking a lot about, for glucose, are we storing it or breaking it down? We're going to look at a lot more enzymes and allosteric effectors that are going to be things like ATP, AMP, just like we saw with glycogen phosphorylase. So when you're learning all this stuff and studying, I think it's really helpful to try to start thinking of everything in terms of, is there a state of high energy or low energy? Is there a state of a lot of nutrients available or few nutrients available, particularly glucose? And this will help you sort of learn why everything is happening in terms of meeting the needs of your cells and your organs, and not just trying to sort of memorize these long lists of effectors and activators, which is, in my opinion, much less interesting than actually thinking about why it's happening. And so I just want to sort of bring it back to this diagram, which was part of the slides, that kind of start to bring everything together. So we talked just now about how glucagon comes from the pancreas. It can talk to your liver. It can start to activate the AC pathway to make cyclic AMP and promote glycogen degradation. Where am I? Yeah, and then we also have insulin. Insulin can talk to both the liver and the muscle, and it can tell them to start bringing in glucose to start making glycogen. We also have epinephrine signaling, and this sort of shows pretty well how epinephrine can signal to both liver and muscle and sort of do very similar stuff to what glucagon is doing in terms of activating that AC pathway and promoting glycogen degradation and glycolysis and all that stuff. So hopefully looking at this can kind of help orient you into how all these things are interacting with each other and how they might indicate different levels of energy or nutrient. And that is, that's all I have for chapter 13. So I know it's a little bit early, but I'm not going to start 14 today. We'll leave that for next Monday. So before I let you go, I guess first, are there any questions about hormones or anything from chapter 13? Okay, so just a reminder. So quiz 10, which is about chapter 12, is due next Monday. Quiz 9, which was about chapter 11, is due tonight if you haven't done it already. If you're choosing to submit a rough draft for your project, that's optional, but if you're choosing to submit it, that's due next Monday, also right after Thanksgiving. No, you have a week. Yeah, you also don't have to submit one. And then this chapter 13, will have quiz 11, and that will be due next Thursday, so a week after Thanksgiving. So for chapter 14, just to give you a sort of summary about it, we're not going to have a separate quiz about chapter 14. Chapter 14 is more of like a transitional chapter where we shift from thinking about sort of properties of enzymes and how they work and more into metabolism and how everything kind of works together from what we learned in the first part of the semester. So we're not going to have a quiz about chapter 14, but that being said, watch the video because it's helpful to set you up for the rest of the chapters we're going to learn. So for next Monday, we're going to do chapter 14 and 15. Chapter 14 is short. And the chapter 15 video is also short, so please, be ready for this for Monday. And have a great Thanksgiving. And I have your exams. If anyone wants to look at your paper exam. And I also have office hours after this if you want to look at them in my office or talk about anything else.